All right, so we're going to continue talking about zygomycota, and this is where it gets complicated. So, we're going to talk about the life cycle of zygomycota. I'm kind of skipping the first phyla because I really couldn't find a lot of information on it. But we're going to just do zygomycota and go from there. So zygomycota, we're going to start with spores, and first of all, I need to describe what a spore is. So a spore is an asexual cell, whereas a gamete, like sperm and egg, that's a sexual cell. And so let me tell you what I mean. In sex, there is an exchange of genetic information. So in the case of humans or mammals in general, what we do is we have sperm. So I'll draw one here, and then we have a much larger egg. To represent the number of chromosomes, we use what's called N. N means how many sets do you have. And so you, as a full-grown adult, you would of course have 2N. The reason you are 2N is because you get a set of DNA from your father and you get a set of DNA from your mother. And so we call those only N. And it's pretty simple. Your mom plus your dad equals you. You have 2 sets of DNA, right? And then of course when you have a child, you're only passing on 1 set. The process by which you pass on only 1 set is a type of cell division called meiosis, and it makes sperm and egg. And the cycle just continues, right? So that's gametes. So then what are spores? Well, a spore is very similar, but it's not sexual. It is an asexual cell, but it does cause reproduction. Spores can grow into entire adults, but those adults themselves will be haploid. So again, with gametes, it is this kind of strategy. N plus N equals 2N. When sperm meets egg, it makes you. But with spores, they are N, and then they just grow into a full-grown adult, still haploid. So the mycelium you see here are also haploid. So we'll start with the spores. Here's a great way to think about it. If men made spores instead of sperm, when they ejaculated and it landed on the ground or somewhere humans could grow, an entire human would grow from that sperm. There would not need to be an egg. However, that human would only be haploid, if that makes sense. The only animals that I know do something like this are honeybees. They can actually make male drones through this process. It's called a renotoky, and that's the closest that I've ever seen a gamete be like a spore. But spores are usually only found in plants and fungus. So anyway, yeah, that's a spore, and it's going to go through mitosis to make hyphae. And those hyphae can just asexually make more spores and just keep spreading themselves, or they can go through sexual reproduction, which is this path here. So sex occurs when 2 hyphae of opposite mating types meet. So there's no gender here. It's not male or female. It's just positive, negative. And it has to do with how they trade nuclei. And so what happens is, because there's no cell walls separating zygomycota, see the last lecture, it's called coencetic division. Because there is no separation, the cytoplasm and therefore the nuclei can flow, and they get concentrated in 2 places. The hyphae will fuse, and that is called plasmogamy, which is the fusion of cytoplasm between 2 cells. This creates a structure known as a zygosporangium, which is how we identify this as a zygomycota fungus. And that's going to be quadrokaryotic, meaning it has 4 nuclei. Those nuclei are not fused yet, and so we have to go through a second process called karyogamy, and karyogamy will now make this 2N. So sex has occurred here. Even though we're working with spores, we have now gone from each one of these being only N, and N plus N is now 2N. And you can see they tried to represent that here with purple nuclei because there was a red and there was a blue and it combined, right? And so then that is going to go through meiosis, actually. And again, remember, meiosis takes diploid cells and makes them haploid again, randomly only passing on half of the genes. And that's going to create a structure called a sporangium. That sporangium begins to fill with spores through mitosis, and those spores are then released. And that's the whole process. To kind of give you an example of, like, to think about humans, if we were zygomycota, and let's say that a guy, we'll name him Tom. Tom meets a nice girl, and we'll call her Cynthia. They meet each other, and they hold hands. When they hold hands, they actually fuse through plasmogamy, and then they're going to go through karyogamy to combine their nuclei, and a sporangium would form. And so sporangium would be like the stalk that grows out. From that stalk, a bunch of spores would collect, and they would be released, and one would fall on the ground, and a brand new child would be born. That is essentially what the fungus are doing. So I'm glad that doesn't happen. That'd be terrifying. Yeah. Anyway, that's zygomycota, and now we'll talk about ascomycota. So ascomycota are actually a very, very common fungi. And just to remind you where we came from here, we've talked about so far Chytridia, which were mostly single-celled, very simple. Then we went on to zygomycota, which were a little bit more complicated, and they had these stalks here that we just talked about. And now we're going to go on to the asco. So ascomycota are most easy to see as cup fungus. This is an example. So they have this structure that looks kind of like this. It's a cup. And all of the spores are made here along the rim, and then all of the sex between multiple hyphae happen down here. Let's see. They are the most diverse of the kingdom. So they're the most diverse phylum. There are more ascomycota than the other phylum. They have perforated septa, which just means coencetic division. They do have somewhat of a cell wall forming between, or a cell plate, but it's not complete. And so that still allows for the flow of nuclei and cytoplasm that we saw before. So we identify them by what's called an ascus, and an ascus is haploid, actually. So haploid means N. So you look at these chromosomes, each one of these is N, but there's going to be actually 4 of them in this spore. And then they go through a process called mitosis, which is how you make your cells. But they're going through mitosis with haploid cells, so they're making haploid spores. So it's just a big clump of spores here, and we call this whole structure an ascospore, an ascus, and then the ascospores are inside of it. So basically, an ascus is filled with ascospores, if that makes sense. And we can kind of see that here. If you look closely, the ascus under a microscope, when we stain for the spores, the ascus is going to be very dark because there's a lot of spores, whereas the single ascospores are lighter in color because it's just one spore. So the ascus makes ascospores, and that's how we know we're dealing with ascomycota and not zygomycota, right? Because zygomycota just makes this like one little clump here, and then the ascomycota makes this long cell, right? So let's look at its reproductive cycle, and let's try to remember zygomycota and sort of compare it. So first of all, if we go back up here to zygomycota, hold on, and I might mess this up. This is kind of difficult to explain. Right, so up here we started with spores. Sorry about the dogs. And then we went through plasmogamy here. And let's see what the difference is. Okay, so the first difference is we're starting with spores. And we're going to do sort of the same thing. Both of these are haploid. So this hyphae is haploid and this hyphae is haploid. And we're kind of doing the same thing as before, but we're making these structures. And I'm not sure why they have a different name, but instead of positive and negative types, we call this Ascogonium and Anthridium. And we can kind of see those here. Really, the reason why there is distinct names for these is because they have different shapes. What happens is one of these is going to give its nuclei to the other. And you can kind of see that here. And this one is going to become the growing one, and the other one is just sort of going to retreat away. And so, yeah, that just has to do with how nuclei are traded. And I can't remember which one gets the nuclei, but it's not really that important for us. Either way, it's going to result in plasmogamy. And so, if we look at plasmogamy up here, we see that after plasmogamy, immediately after we went to karyogamy, and then we sprouted. The difference here is we're not going to karyogamy immediately. So before, in zygomycota, these would immediately fuse their nuclei, and you would get the purple nuclei, right? Here, there is no fusion, and so we're just going to go through mitosis and create two branching hyphae. This creates this cup shape known as an ascus, and it's why the fungus has a cup. And so these are dikaryotic because they have two nuclei, and both of those nuclei are haploid, right? And so the nuclei and the ascus fuse afterwards. So after this whole ascus has formed, then we go through karyogamy. And so just to compare, in zygomycota, what we did was we went through plasmogamy, and then we went straight into karyogamy. Whereas here with the ascomycota, what we did was we went into plasmogamy, and by the way, after that was the sporangium. This time we made the ascus first. So we grew the reproductive structure first, and then we're going to go through karyogamy. So it's just a slightly different process. So now, after karyogamy, we are 2N, because we have just combined two haploid nuclei to create this. Then, we're going to create this ascus, because remember, we're fusing two of these cells here. So there's two nuclei there, and we're combining them. So that should make 4, I believe. That's not right. I guess it went through mitosis or something. Either way, it goes through meiosis now, and that's going to create all of these, which are spores, and then those are dispersed, and the cycle continues. So the only real difference here is when they did karyogamy, and also how they combine their nuclei with this strange structure. Either way, this is sort of the cycle. So down here is where sex occurs. So we'll get like one fungus coming in this way, and then we'll get another fungus meeting it over here, and they're going to sort of fuse here, but they're still, how do I draw this? They're still not going through karyogamy, so it's just like both hyphae growing together, really. And then they go through karyogamy at the rim, where they're making all of those spores come out. So you can kind of see it here. Dikaryotic, but then it creates these hybrid karyogamy nuclei, goes through meiosis, then mitosis, and then releases the spores. So, yep. Basically, what you need to know from here is sex happens down here, and then spore creation happens at the rim. And again, there's those ascus. Here's another good picture. So, again, we have the positive and the negative mating types. Each of them is only got one set of DNA. They come together. So I'll do the whole blue thing. So we'll say this one is the blue, and this one is the pink. They meet, and they sort of form this fusion. And then you get these dual hyphae with that are dikaryotic growing up. It's just like two hyphae growing together, honestly. And then you have karyogamy, which means the nuclei fuse. And then you go through meiosis and then mitosis to create all of these spores along the rim. So, yep, that's Ascomycota. Now to the most edible of the group, the Basidiomycota. So they are also called the club fungus. This is probably the one that you're used to seeing the most. They're the most edible fungus, so very, very fun to have knowledge of. And they sexually reproduce more frequently than asexually. And here's how they do it. So again, mycology is all about reproductive strategy. So let's go over this one. Okay, so we're going to start, just like before, with the spores. And let's see, I haven't actually looked at this in a while, so give me a second here. They're going to grow, right, through mitosis. And, ooh, it looks like these actually have complete cell walls here. So these are normal cell walls. So then, obviously, there can't be more than one nuclei here. So we're going to have a fusion here, plasmogamy, just like before. And then what's going to happen? Resulting in the formation, okay, dikaryotic. So that's a lot like the ASCO. So we have the same thing happening as the ASCO. So we have these dikaryotic nuclei. They're not fused yet. And then we just make the whole reproductive structure. That's correct. Yep. And then we make a spore. Okay. And then we make a basidium. Okay. And then cell division. All right, so pretty much this is the same thing as Ascomycota. I'm trying to see where the difference is here. Can anybody spot the difference? I think it's going to be like right here. So let's see. We've got a zygote, which is 2N, and then it becomes basidium, and then up here, let's see. We had this, and then it's not a big enough difference to really notice. So basically, the reproductive strategy is almost identical to Ascomycota. It just makes a different structure overall. So, yep. And it makes what's called a basidia spore. So the basidia spores are different from other spores in that they are released with water droplets. So this is called Buehler's drop. It's kind of cool. They have this little water droplet they always keep on them. And then when another water droplet falls into those gills, it's going to fuse, and that's going to create a rapid shift in momentum, and that will release the individual spores. So that is why it's different. Even though the reproductive strategy here is very similar, here is where it's different. Okay, so that is the edible ones. Now we're on to Deuteromycota. So this is actually not a current phylum. So this is asexual reproduction only. Recent DNA studies show that members are not monophyletic, meaning they belong to other clades like Ascomycota and Basidiomycota. And so this is just a description now. It is not actually a real classification. But such members include penicillin and cheese mold. Okay, so here is an actual clade. This is the newest clade grouping. It's called Glomeromycota, and one of my favorites. It's a new phylum. They have coencetic hyphae, just like the Zygomycota, but no zygospores, so not quite Zygomycota. They do asexually reproduce. And then they have what are called mycorrhizal associations, which is really neat. So these fungus, let's say this is the fungus, what they can do is they can actually connect two different trees with their hyphae. And they trade nutrients with the tree. And this can actually create an information highway between the different trees. Glomeromycota are extremely important in the growth of plants. Without them, plants just don't grow. It is an extremely necessary part of our ecosystem. What's even cooler is these fungi can not only trade with the individual trees, but let's say that this tree has a whole lot of carbon and this tree has a whole lot of phosphorus. they can actually trade between the trees using the fungus as a middleman. And then, of course, everybody knows if you're the middleman, you skim a little off the top, and that's how they sort of make their living. Anyway, that's all I have. Here's some stuff if you want to look at it. Hope you liked it, and I hope the reproductive stuff wasn't too confusing. Basically, what I would take away from the whole reproduction stuff is know what plasmogamy is and know what karyogamy is, and you should be good. To be honest, I struggle with this stuff too, I'm not going to lie.